So this is a talk about a 3-year study that's actually years, and it is prompted by a new plant that was built on the west coast of West Australia. Now there are 6 desalination plants around the southern half of Australia. They're all big ones, and the one that we were dealing with was just this one here, about 200 kilometres south of Perth. And state of the art, uses reverse osmosis. But what is reverse osmosis? Well, it's the process of forcing seawater through membranes, and you end up with absolutely pure water, no contaminants, no viruses, bacteria, hormones, heavy metals, just water. And governments love this because you can see the best, the most efficient, the everything. But it has one downside, that you end up with 60% brine at the end of the process, and you have to do something with it. And what's in this brine? Well, salty water, which can be anything from 50 to 70 parts per thousand. So normal seawater, that's almost double seawater. And there's also lots of chemicals, which, and these are things like coagulants, anticoagulants, biocide, things to clean the membranes. And you see the whole list here, but it's not, some of them aren't very friendly chemicals. Now, most of the desalination plants are on the coast, so they're disposing of that effluent into the near shore. And old-fashioned desalination plants used to boil the water, so it's hot water coming out. But the new ones, the reverse osmosis, there's not really a change in the temperature very much, so the water tends to spread out over the sea floor, depending on the density and the temperatures. And this massive salty water can pose a threat to the benthic fauna and, of course, seagrasses. And one of the ways to get rid of this is through big diffuser arrays. So here's one of the risers from a diffuser array in our first desalination plant in Coburn Sound. And they were testing it with rhodamine dye, which is why it's pink. Usually it's not. And you can see it shooting upwards, and behind the hulking diver there's some dilution going on, but it may not be all that much. So the desalination plant we were dealing with was going to be releasing brine into an area called Geograph Bay, which has got vast seagrass meadows, about 120 kilometres of them, and they go down to 50 metres deep. And here are some of the environments that are in somewhat shallower water. So you've got, oh, don't do that. You've got Posidonia, australis and amphibolus. You've got reef pavement with these big sponges, great big things like this. And here's one of our quadrats. So the question that we were posing, would this desalination discharge harm the benthos, the environment, etc., etc.? So we used 2 approaches to answer this. First of all, a huge benthic survey along 70 kilometres of the coastline, and there were 8 sites, 6 transects at each site, 5 quadrats of a metre collecting benthos, invertebrates, 6 seagrass quadrats, and this was sampled before and after the start of discharge. So I'm just going to show a really brief thing of this. There was an effect near very near the desalination discharge point up here. So that's the red arrow on the seagrass. So the density was lower and the plants were smaller. But was that due to all the disturbance that was going on during the construction of the pipeline, because they had to blast it out, turbidity, all sorts of things going on, or was it due to the discharge of brine? So not a clear answer from that. And we also did a multivariate analysis for the invertebrates, and a very big study, and there were differences in the invertebrates near the discharge site, but so that was the first year. The second year, when we came back, there had been huge winter storms. This is a really wave-exposed coast, waves up to 8 metres high during typical winter storms, and the sites near the discharge point had been covered with 1 metre of sand. So rock pavement with invertebrates and seagrasses became sand, which is a big problem if you're doing a multivariate analysis. So our conclusion from that was that establishing causal relationships between brine discharge and the environment was very difficult. So we took a second approach, which was to look at the brines and salinity under control conditions in a series of mesocosm experiments. So here were the first ones in tanks, and we looked at all of those things: survival, growth, photosynthesis, osmoregulation, water relations, ion accumulation, and osmolytes, so sugars and amino acids. And we did this for 2 weeks, comparing brine and salt water. And this was to answer the question, is brine more damaging than the equivalent salinity? So the brine comes out at 54 parts per thousand. Is salt water that's 54 parts per thousand, does that elicit the same response? So to do this, we standardised our responses to the controls. So what you can see in this graph here, so this is just growth, leaf growth per day. The red is just the salinity, so these are the controls here. And then 46 parts per thousand is equivalent to 50% brine, and the 54 is 100% brine. And you can see that there was a huge response with the brine, and quite a bit less so with just the salty water. The leaves there are showing signs of salt scorch. With the next one, same setup, results percent change standardised to the controls, and you can see that photosynthesis, not much response for the 50% brine, but quite a big response at 100% brine. And osmoregulation, this is the, I'm just showing osmotic potential. We measured other things for this, but this is the increase in osmotic potential, which ensures that water does not flow out of the cells and dehydrate the plant. And you can see that there is a significant response at 100% brine, but not really at 50%, which is 46 parts per thousand, so it's pretty salty. So the answer to the question, are the effects of increased salt and chemicals in brine additive? And it's a big yes. We also tested Posidonia in just salty water, so that's increased salinity, for 6 weeks because we hadn't had a lot of response at 2 weeks. And this has already been published in that paper, so I'm not going to go through the results of those, but just say that the responses were dependent on the salt concentration and the length of exposure. And my last one are little seedlings, which we put into brine for 50 days. And you can see that the ones that were in 100%, and then we put them in recovery for 20 days. And so this photo shows after 20 days recovery, and you can see the ones that were in brine for 50 days, they even started growing again. So they are one tough plant. And so we did all these things about biomass. So the conclusions to the study were that the discharge was probably small, the footprint was probably small. Brine is more than salty water, and seedlings are very tolerant. So if we take that to management implications, and not in any particular order, regulators want to be able to put things in our state in 3 zones. So we've got the high impact zone where quite a lot of change is allowed, the medium and the low. So this is an old diagram from the Coburn Sound plant. How far, a reality check, how far from the outfall would you find 50% or 100% brine? How would you relate our results to real discharge? If you were looking for biomarkers, what would you choose from the experiments to inform the regulator about what to look for? And I'm still thinking about that, so I'm leaving it as a question. Right. You're on time.
Thank you, Marion. We do have some time for a few questions, so go ahead. Yes, please.
Yes.
Yeah, I have one or two questions. Did you observe more calcifiers in the seagrass meadows exposed to the brine or not? Theoretically, it's possible. Would agree with
I don't think you could tell. It's highly variable, the benthos there, and like many of the things in Australia, they're growing in a low nutrient environment, and you get a very strong replacement of species over short distances. I think there's corals and sponges and mollusks and calcareous algae, and the sediment is a mixture of recycled silica from the land and calcium carbonate generated in situ from the fauna. So it's not as high a calcification as you get further towards the tropics, but it's still a lot. So I don't know if that answers your question, but the range of species was immense, so yeah.
Another question?
Yeah, thanks, Marion. Really love that talk. I was really buoyed to see that the recovery of the exposure after 50 days was there.
Mm.
But it jogged in my mind the possibility of, would they get that option in the desalination operations? Do they give a break of pumping out brine to get some potential recovery, or would they?
I think they do in Coburn Sound, and it's not because of responses to salt, but it's responses to deoxygenation, but there's no seagrass in the Coburn Sound area, bit where they dispose of the effluent. I think where they've put the plant at Binningup is probably the ideal place to put one because of this massive dispersion, and it's really different to Adelaide, where it's going into very calm waters. I'm off the hook.
Okay, unfortunately our time is finished. Let's thank Marion again.